Please, everyone rename yourself so we can know who you are. And one more thing I didn't mention yesterday is that we will check your attendance for both days, yesterday and today. And then if you attend both days and finish the whole session, GBIF will give you a certificate after the workshop. Okay. Okay, so please stay with us for the whole sessions. Okay. And okay. Let me have a quick review for you yesterday. We just have a introduction to gbif.org, so you learned how to I think you already learned from yesterday how to filter the data you want to use, the gbif mediated data, and know about the common data issues you might be aware of. And today, we will learn about the API and RGBIF. Then you will know how to use R to dealing with the data issues we mentioned yesterday to improve the data quality for fit for use. Okay. And today, actually, we have another new trainer. Is Daphne Ho. So I just let Daphne introduce herself.
Hello, everyone. You guys can hear me quite clearly, right? Okay.
Yes.
I'm Daphne. I am from Malaysia, but but most of my time right now so far I wasn't working in Malaysia. So I did my PhD in Taiwan, in biodiversity. After that, I became a postdoc in GBIF, which is the Taiwan node of GBIF, for the while. And now I'm currently an ecology postdoc in China, so one of the university in Shanghai. Um, I am also the Asia representative of Hedwig, which is the organization that maintain and design Darwin Core Standard. And yeah, so for these few years, I am active GBIF trainer, mentor, and also translator for their documentation. And I will be guiding and teaching you guys about RGBIF later. So nice to see you. Thanks.
Okay. Thank you, Daphne. And okay, let's just start for the first session. VJ, is your time for introducing the API.
Okay, thank you. Let me start my screen sharing. Can you see my screen?
Yes.
Yes. Then you can hear me well?
Yes.
Okay.
So far, so good.
Yeah. So, today in the first session, I'm going to cover an introduction to the GBIF APIs. So, a lot of us may be wondering what API is. So, what is GBIF API? API also also called as application programming interface. So it's basically interface from which computer programs can talk to the GBIF website, to the GBIF data servers to get the data. It is used by different clients, like you can use R or Python or Perl or different kinds to
VJ,
talk
VJ,
to the GBIF API. Yes.
Sorry, VJ, your sound is like voice is breaking. Just after you check if we can hear you well, then it's breaking.
I think it's working on
Oh,
and off.
you are not able to hear me now? Oh, okay.
Now
Yeah,
Just
it's
give me one
okay.
moment. Oh,
now
okay.
it's okay.
If it happens again, just stop me and let me check if I can
Okay.
do something different. If it is working right now, that's all right. So yeah, we can use different clients to talk to the APIs. It's a problem again.
Yeah. Every time you say
Just give
okay,
me
it's
one minute.
fine, then it start breaking.
Yeah, yeah. Give me 1 minute.
Okay. Okay, when we are waiting for VJ, oh, just to make sure you already like watch the video on about the GBIF introduction, API introduction. So here is the link, and that what VJ will talk about. The introduction is almost similar or the same as the video. So if you have not uh take a look yet, please just have a look.
Can you hear me better now?
Yes, it's better now.
Okay, let's try. I actually plugged in cable into my.
Um, VJ, maybe you can just turn off your camera. I think like it was fine when you turn it off.
Okay, is it better now?
Yes.
Can you see my screen and hear me?
Yes, both are
Okay,
fine now.
okay. So let's hope it works now. Yeah, so GBIF API is basically allows us for more refined queries and automation. And
The voice is breaking again, sir.
It seems like you cannot like speak for more than three sentences. Um,
okay, what is happening? I'm not sure. Okay, give me 2 more minutes. I will try something else and then if it doesn't work with you.
um, okay. If it still happen, maybe I'll play the video instead later. Or can anyone like give me thumbs up if you already finished watching the video yesterday or today? API introduction. If you did, please give me a thumbs. Okay, it seems like only 2%. Okay, 3. Okay, 4. And if it's not that many, then I think I'll try to play the video. Let
Yeah.
me check.
Let's see if it is better.
Yeah,
Okay. I will try to talk slowly, maybe.
okay.
Take pause it.
Slower is better.
Yeah. So GBIF API has 5 different namespaces or entry points. We can access the registry, the species, or the taxonomy, the occurrences, maps, and literature. So what exactly is API? API we can imagine as a totally different scenario. It's like a restaurant. A customer goes to the restaurant, uh, then he needs to communicate with the waiter in a language that excuse me, the waiter understands. And then the waiter is actually the interface between the customer and the chef. So once we convey the order for food to the waiter takes it to the chef, and chef creates makes the food, and uh, the waiter again brings it back to us and serves it. And then once the waiter is back, he will again convey, okay, this is this soup that you order, this is this dish that you order, and then the consumer consumes it. So APIs are very similar to that. So imagine the customer as a user, and the client or the interface is the language that we both understand, which is Python or R, or the user interface in gbif.org. The next step is when we tell what we want to do, we talk to API. APIs take it to the GBIF data server, brings back the data, and presents back to us. And then depending on what we want to do, the gbif.org website or our R interface will slightly rearrange that and present the data to us in different formats. So this is how the APIs actually work. Oh, sorry. Okay, so in reality, when we look at APIs, the colorful line on top is what we call as a API call. So in that, we are telling gbib.org that from version 1, I want occurrences, and I want to search it for a particular taxon key, for a particular country, for a particular year, and things like that. So this is how we describe what exactly we want in terms of API. And then in the bottom, whatever you see is the output of the API. Now, if you just look at this output, it looks very confusing and cluttered. So let us try and break down the output. So this output returned and or what we are looking at is in a format called JSON or JavaScript object notification. So let's learn a little bit about the JSON format. So JSON format essentially is a key value pair. So there is a key called name here and the value is Vijay. So this is how the JSON really basic key value pairs work. And then to describe in a little more complexity, they are saying height is the key and the value, there are actually two different things. One is the actual value which is 178, and then there is another key value which is pair which is a unit and centimeters. Because if I just say height equals 178, we will not understand 178 centimeters or inches or meters or what it is. So we can keep on adding and keep on nesting these key value pairs to make a complex structure to pass on the data. JSON also supports arrays or lists. So this is a list of things that you can pass uh, when you are passing the list, it is in square brackets. When you are passing the key values, it is in curly brackets. So to kind of put it all together uh, in a little more complex structure, name is Vijay, hobbies are eat, walk, and sleep. Height is 178 centimeters. So this is, we can keep on building. We can put more, we can say name, we can say family name, we can say country of residence, whatever we want to add. We can keep on adding and basically create a complex structure. This is the language that the computer programs speak to each other. This is one of the languages. So JSON is one of the language standards. So now that we have understood JSON, let's go to the uh, uh, to understanding little more about the GBIF API. So the API types are there are different types of comparison API. Get is to access the data. Post is to send the data. Put is to update the data, and delete is to delete the data. Out of which post, put, and delete would require authentication. Just getting the data, anyone can access. This is how GBIF APIs are set. For different APIs, it could be different, but for GBIF API, just to get data, there is no authentication required. But to post, put, and delete, you need authentication. So that is how the APIs are structured. Going to a little bit more details about API, you can go to this particular page, gbif.org/summary, and you will get all the details about the API and very detailed documentation. So if someone wants to understand them and explore different APIs, this is the place you need to be visiting. As I already mentioned, there are five entry points for querying APIs. There is a register entry point, which is give you information about data sets, organizations, IPT installations, and things like that. There is a species API, which gives information about taxa or checklist or names like those. Occurrence database will actually give us occurrences and statistics. There is a maps API, and there is a literature API to find out who is citing your data. Out of these, most of the times we use the occurrence or the species API. Now, getting into little more details of how we use the API. So API call structure is typically you start with the HTTPS and the base URL, and then you tell which API you want to use. Within the API, there may be several functions. So within that, we will say which function we want to use. And then after that, we will say what parameters we want to query. So this is the base structure of the API call. And looking at the actual API call, we will say that we want to access the api.gbif.org version 1. And then the API we want to call is the species API, which is the taxonomy API. And then the function we want to call is match. And we say we want to match the name person. Castor, if you know, is the genus for the common sparrows. So this is a complete query that we can pass. And if you type the second line in your browser, you will get some result, some text. What we will get is something like this. So this again, now when you look at this, you know what it is. It is a JSON, and in JSON you'll say, okay, the usage key is this, the scientific name is this, the canonical name is this, and rank and status, everything that you see here. Now you can understand this better. That if you type this, this is what you are going to get. So these are kind of the inside of the APIs and how we basically try to use. Now we can make these API calls more and more complex. Like here we are saying from the occurrence, this is the taxon key I want, but I want the records only with license CC0-1-0, and then I want only the records with occurrence status is present. So this is actually the GBIF URL. If you open gbif.org, go to the open search, and you are selecting certain parameters, this kind of long complex URL is what you will see in your access, and the same URL is actually interpreted into this API, and actually in the background the JSONs are being transferred between whatever user interface, that is your website, gbif.org, or your RGB for 5G, and the GBIF data server. So this is the API call, and if you see all the text in the bright blue down here remains the same in both. Only thing is the access point. Where here we are using it from gbif.org occurrence. Here we are using API website occurrence. So this is how gbif.org is also using the APIs in the backend and showing us the data. This also lets us use these things to build our own API calls. Okay. So now if what happens if there is a lot of data? So when there is a lot of data, we need to browse through pages, because if we send a query for something like a sparrow, there will be millions of records, and our website or our R will get stuck just getting all the data. So what GBIF does is GBIF sends the data in pages, so that GBIF server is also overloaded. So here, if you see, it says that limit is 300 and offset is 1000. So what it tells is basically give me only 300 and start from record 1000. So this is how it will. You will have to add it in the query, that limit and offset, and then once you have those 300 records, you can process those and then fetch next 300 records. So that is how the pagination works. So if you look at the JSON in detail, it tells you the offset is 1000, the limit is 300, and is it the end of the records? No. And in count, it is telling you how many records are there, right? So this is the paging information, and this is the total number of records available. So you know that you are only between 1000 and 1300 records. There are 25,000 more records still present. And in the bottom, it gives you all the details, like for which species it is and all these details. This is how the whole big complex GeoJSON is broken down and understood. So now that we understand how these things work, let's get into a little more detail. So This is pagination, and this we are going through pages. But if you want to do asynchronous, that if you don't want to have any limit, you want to get bulk of the data, then you have to use the occurrence download API. This also allows you more complex queries. And the best part of it is when you are downloading the data using this, you also get a citable DOI, which you require for any research. It saves the downloads in your memory. It uses post method. And if you remember earlier, get method doesn't need authentication, but post method will need authentication. You need authentication, which means you need a GBIF account. And if you have carefully read the email said that you make sure that you have registered with GBIF and you have your own account so that you can run these queries. So the occurrence record you authenticate with using your GBIF account and use some kind of client. It could be curl, it could be RGBIF, 5GBIF, or you can even use the GBIF website which you have already made up, used and downloaded data. So these are the different ways that you can access the open download API. And then just to give a crude example, in curl you need to include the username and password. You need to tell in the header that you want the data in JSON, and then you actually pass the query in query.json file, and then you call it using the curl request. So if you go to this particular website, if you are familiar with JSON, and if you are brave enough you to play around with JSON, this is a good that you can use. You can go here and write queries in JSON, and then you can get the data download. So to create a query in JSON, this is how you create. You say create a username and address. All the details you have to specify in the header, and then you have to specify the predicates. Predicates are nothing but the query parameter. So what are the different kinds of predicates? We can use predicates like equal and like. So this is equal to that, or this is like that, which is like a wildcard search. You can obviously use and or to build upon more predicates. You can also use not if you want to say not this, not that. You can in and give a list of things. You can use greater than or equal to, less than or equal to predicates. You can also use within predicate if you give the list. For geographical queries, you can use geodistance predicate. You can also use is null and is not null to just saying okay, these values. If the values are null, I don't want that data. Or if the values are not null, I don't want the data, kind of thing. So you can use those predicates too. So just an example of in predicate, you can use in predicate and then give a list of taxon IDs. So if you, in this example, if you give these 3 taxon IDs, they will kind of map to these 3 particular scientific names. So this is how you can run a query for multiple taxons at the same time. You can say, I want this and this, all the data. And then you can also say that I don't want any data that has here and null. So there is not null here. So that you want only the data which has year specified. So this is how you can go on building a little bit complex query. And then you also say that I want to get rid of all the records with Z. So if the recorded date is invalid, I don't want it. If the matches for Z, I don't want it. And if the taxon match is higher rank, I don't want it, which means I want only the records with these three particular taxon ID at species level identified record. This is how you go to that, and then you can just actually copy paste the query into that interface, and then you can get the download. You can see the query whether in a kind of graphical format. Then you can actually use one of the buttons to download that data. So this is how the occurrence download query API works. The whole purpose of explaining you this is for you to appreciate how complex GBIF data processing is, but also to understand the nuts and bolts of writing this complex query so that you can actually extract data as much close to what exactly you want. So this is about the the GBIF API details. I know it's very technical and complex content, and I'm happy to try and answer any questions.
Thank you, VJ. I think it's also technical issue to hear you well, but I think like they can also, if they did watch the video and check all your slides, I think maybe like everyone can understand the most of it, I hope. And now it's like the QA time. So if you have any questions about what VJ were saying, you can ask it now.
Melissa, Vijay, this API, the calls you are talking about can be directly put onto the API interface and get that result. That whatever you has written those API calls. And what is the need for R Studio here? Just asking. R Studio.
Yeah, so with R, we can automate these things. We can write a script to run through different different species or different regions and get that data one by one and process it on that.
I can try to answer for this because Vijay's voice is breaking. If I understand your question correctly, the function of API is just another way for you to download your data, and it's especially convenient if you have a lot of data and a lot of different filter to do. So for the R that you mentioned just now and what we are going to discuss later, it's more on we are using R code and R script and R interface to access GBIF API. So the R team, they developed this RGBIF package. This package already written the function of wrapping this API together inside the R package. So we can directly just use the R function to download GBIF data using the API that they have already designed. So I hope I answer your question. And then in R as well, we can use the function that they design to do our filtering. Depends on what kind of data that you want.
Thank you. I could understand that there are ready-made functions written using the scripts, and this will just
Yes.
invoke the function and you get the particular API script running in GPU. That you mean to say for getting a processor
Yes.
query.
Yes.
Okay.
The R code and the R function is already wrapping the GBIF API
Yeah.
inside of it. We are still downloading GBIF data using GBIF API. Correct.
Is there a documentation of how many functions are there in the TAR package for GBIF?
You mean for the API?
That package which you have downloaded and installed just now, is there a
Oh,
documentation for
okay.
that?
Yes, I will also show you the documentation and the link later.
I think you can also download it from the course we provided.
Yeah, the training
Yeah.
material link has most of the content that we teach for these 2 days.
So the one I pasted is the file for downloads. You can go for the R file. If you scroll down to the bottom. So that would be the most script you will practice it in the next exercise. Okay. So Aditi, you have question?
Yeah, I just wanted to just clarify, since I'm not from the basic background where we code and all. So we are just trying to understand how the functions and the search filters work on GBIF. This is the purpose, right?
Yes, and I
Yes,
will
that's
guide you
what.
later. And
So we
because
don't
I'm
have to
also
actually do this. This is just to, yeah, definitely.
Pardon, your voice were also breaking.
can you repeat your question?
Yes, Aditi, your voice were also breaking.
Yeah, the thing is that we don't have to actually do these things. We just have to understand the background of it. This is what is expected, right?
Yes.
No, but
Uh, uh,
yeah. Actually, in the exercise, we will have like simple, script for you to let you know how to, get your data from the API without go to gbif.org itself.
Okay, okay.
so what VJ has mentioned um, is he was teaching about the basic structure of API and how
Yeah,
the
yeah,
filtering will works on the API URL. Mm,
yeah. Okay, yeah, I got the point. Thank you.
And
So in terms
Sabina,
of
yes.
in terms of really using, because I'm also a heavy R user, I don't use GBIF API on the web page that much. So it depends on your purpose, and if most of us here are using GBIF data for ecological related analysis, then maybe we will use R more than the one that Vijay just mentioned. Mm.
And I
So,
want to
so we will
emphasize.
be needing these APIs for working on R.
Yes,
Yes.
but you don't have
Okay,
to write
okay,
the
okay,
structure like what Vijay just showed, but then in the exercise later we will try to do the basic.
okay, okay.
Mm.
And like I just want to add something like not only for R, like if you already, some of you, I think you already like attended the data mobilization workshop before, and we introduced about the OpenRefine. Is also the
Yes.
last one is also using the API from the GBIF, and you just put the API script in the OpenRefine, then it can
Okay,
track back the data you
okay,
want. So the
okay,
API is basically is not from you to design it, but like the website side. So gbif.org, the informatic teams will design the API to let user use what kind of data or field they want to provide for users. So they will decide it in the API, and then you can just uh extract the data
extract it.
from the
Okay,
API they provide.
okay. Yes, I got the gist of it. Thank you, Daphne. Thank you, Melissa.
And Sabita also had a question. It's like, is is it much faster from GBIF API than from within R to filter and download huge datasets?
Um, for Sabita's question, I think it depends on your purpose. So um, when we download data using R, it's because we need the data to do other further analysis. For example, we want to visualize the data and we want to plot it on the map. So that is a preparation and it's more convenient already when you use that directly in R and then for the further any kind of data analysis you would like to do. So you can also download it from GBIF API if you don't use R for your data analysis. So this really depends on your purpose. So some people, for example, some tools or website developer, they use GBIF data, but they don't use it for data analysis. So it's actually more convenient for them to extract data from GBIF API directly. Yeah, so I hope I answer your question. So it depends on your purpose, but both of them and any of them uses GBIF API to download data as the foundation to download. Yeah.
Um, maybe I can like explain it in another way. It's like basically the simple way to explain what this API is. The API is just like a menu when we go to the restaurant and you want to order some food from you will check the menu and then you will order the food to the to someone and then that menu is kind of API. So the restaurant will provide the menu and give the dishes and the food they are providing, and then you can just choose from it. So that's why API is called the interface. So it's just kind of the menu you want, and you can get the food from by ordering from the menu and do and get the data. So it's the food you will get. Okay, I hope it will be more clear for that.
So using in R is very helpful when you want to do the same thing again and again. Repetitions become easy. You set the script and then you just run it anytime you want it. You change the name of the species, you run it. You change the region, you run it. So that's the advantage of uh, using R over doing it manually, taking the data, putting in Excel, plotting the map or things like taking it into QGIS. If you can want to automate it for multiple regions, multiple species, tools like R are used.
Okay. And yep, I think you will get like more familiar about the API when we go to the next exercise. It's like finding taxon key. But I'm not sure, VJ, is that okay for you to 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 explain about the exercise?
Yes, I will do
I
it.
hope the sound is good enough.
I think you can try to speak slow.
Yeah,
Yeah,
I will.
slow or
Mm.
make some pause. Like it would
Yes.
be better if you.
Let me start my presentation.
Sorry, VJ, are you still there?
Yes, yes. Actually, for some reason it is. Uh, can you see it now?
It's still
Yes,
running.
yes,
Okay, yeah,
yes.
we can see it now.
So, basically, when we are querying GBIF APIs, in most cases, it is very important to get the exact species or taxon data. So we will go through this exercise to learn how to understand and try to fetch data for exact taxon. So some concepts: taxon keys are issued for every taxonomic entity, that is species, genus, family, order, and so on. These are the unique identifiers issued for every accepted name, and for synonyms, they are linked using the accepted usage keys. This allows user to ensure that they are collecting the data for the right species. And to facilitate multiple species download, also you can just list these numbers. So taxon keys can be found on either gbif.org. We can use species API, or we can use the species matching tools. So so what we can do is we can use the species API to get the taxon keys for these 4 entities that we have listed, and then we can compare the results. So for getting that, getting to the species API, we go to the species the species endpoint. I will try to paste the link in the chat. If you remember, I used this one to illustrate the example, right? So what you have to do is copy this URL and try to run it. And once you get some data, try to interpret, and then in place of the parameter name equal to pacer, you copy paste these, you type in these 4 names. I can also copy paste them for you. Put them one by one and then see what results it gets, and note down the values that you receive and your interpretation. Is it clear? Could you hear me well?
We have to
Not
put it on
really.
the API, gbif.api window.
No, you have to put it in your browser top window.
Okay, that on the address bar.
Yeah.
Okay,
Not
that
in
what is the
GBIF.
link?
So
Can you share the link?
I've shared the link, right? You see it? Or you want me to share it again?
Yeah, the link
Yeah.
is already in the checkbox.
Yeah. So first try to run that, and let us know if you are able to run it and see something, some text at least.
Acha, I am getting some that on JSON value.
Correct, correct. Now we have to interpret it.
Okay.
Yeah, even I am getting it.
Yeah. So now what you do is you have to run it for the values that you see on the screen. If you want, I can copy paste them.
Yes,
In the
please.
chat
Yes,
window.
please.
Yeah, just give me one moment. Here are the 4 values. Now we have 5 minutes you copy paste each of these values in place of pacer. So name equals lepus saxatilis, name equal to aves, like that you run 4 times. Try to look at the values and see if what is the taxonomic status it is showing.
Sir, when we are mentioning the name, do we need to use the author name also for the
Everything, yes,
species?
everything
Okay.
complete.
Yes, all you
And
need to do is copy paste on the URL link that VJ provided.
yeah,
And it's like name equals something. So the something, the parser is the value you need to paste.
yes,
Yes.
that is right. Now you try to interpret what is the taxonomic status from this.
Okay, from the results we have,
Accepted.
from the results
Status
we have to,
accepted.
Yeah, correct. So that is what
First
you have
one,
to
the
check.
status is accepted.
Correct. So that is the first one. So
Yeah.
try it for all 4.
Yeah. Okay.
so just the status we have to check.
Yeah, that is what we are trying to do right now. I mean, there are so many things that we can interpret. Right now, we are just
Yeah, that is what I was actually wondering.
Yeah.
Yeah.
Right now, we are only focusing on status.
So for the first one, it is accepted. For the second one, it's accepted. For the third one, it is I don't synonym, and for the fourth one, it is
Magnolia
doubtful.
phyta is synonym, yes.
Correct. So, was everyone able to do it?
Yeah.
Yes.
Anyone who's not able to do it?
Yeah,
Anyone
we're
who's
not able to do it. Please
well?
raise your hand,
Yeah,
and we can help you to do it.
exactly.
Why
then
is it considered doubtful? It can either be accepted or a synonym.
Yeah.
No, there are many status for different kind of taxonomy because the names are keep evolving, right? So it is acceptable to have synonym, doubtful, accepted, etc. There are different kind of categories.
So when if we are using a name which is not the most recent accepted version, at that time we get the doubtful as the result.
But
When do we get doubtful?
then it should be a synonym if it is right now
Yeah,
not accepted.
correct. It should be synonym.
Yes,
When
then
do
why,
we get doubtful?
exactly,
I mean,
that
we
is
do
my question
understand
to you.
synonyms. When do we get doubtful?
If there is any controversy or if there are 2 competing groups trying to prove things.
Okay, understood.
Okay, yes.
Yeah.
Okay. Yeah. So when the species status sometimes is not very clear or not very taxonomically resolved, in such cases we
It will
say
be
that it is
doubtful.
doubtful. Am I right?
Correct. But GBIF
Yeah.
still
Okay.
needs to list it because someone has it in the literature or someone has it
Right.
in the database.
Right. Can we try our own these things
Yeah,
meanwhile?
obviously. Is there anyone who was not able to run this?
Or do
Everyone.
I need to also share my screen to, if you want,
I mean, I can do that, yes.
okay. Yeah, maybe do one for them to let them know more.
Okay. You're able to see my screen, right?
Yes.
Okay. So here what I need to be doing is I will just copy paste the link that I provided, okay, in the URL box. So this is the,
Yes, sir.
so you can see my screen output.
Yeah.
Yes.
Now you are wondering why it is looking different than your screen. Are you seeing the same or you are seeing it slightly different?
We are seeing a text. You are seeing it as a JSON, Pakka JSON
Yeah,
value.
yes. We are seeing it as a text.
Yes.
Right. So the trick is I have added a JSON formatter
Okay.
add-on to my browser
Add-ons.
because
Okay.
because
Okay.
my day-to-day work involves a lot of JSON reading.
Okay.
And if I'm seeing it as a just text, it becomes very confusing and it takes a lot of time. So if you
Okay.
want, you can try and get that.
I am getting an option of a checkbox called as pretty print, and if when I click on that checkbox, the thing is text is converted into JSON.
Exactly. So some tool like that is helpful if you are going to do a lot of JSON.
I also added it to the my
Okay. So
Chrome JSON formatter. Now it is showing the way you have.
yeah, exactly.
Yes,
Yeah,
yes, me
exactly.
too, me too.
That makes the life much easier.
Yeah, that is easy to read.
Yes,
Because
yes,
I
yes.
was or else looking for it. It's difficult to see it as a text.
Now
You may
what I have
visit
done is
the three dots at the end and then click the extension and search for the
right.
visit Chrome web store and get the text.
Yeah,
It is only
I'm
on
using
Chrome. I think it's
I'm
only on
using
Chrome.
no, I think there is another one available on Firefox also. I don't
Okay,
know
okay.
about. There should be something
Different
available. Edge also.
JSON output is quite commonly used within the
Mm.
developer. So no matter any browser you're using, I'm sure there will be some JSON pretty viewer extension for that particular browser. Mm.
So now what I have done is I have replaced the pacer with the name, and when I copy paste that, it automatically adds this
Yeah.
$20 for spaces, just to make sure that it understands, and now it gives me the output. And here, what I'm supposed to be looking at is the status and see whether it is accepted or not. Correct? So let's go back to the slides. What I have done here is basically copy pasted the output to see. So these two are accepted, and this is a synonym. And this one is doubtful because there is some problem with this particular name. Could be when it is doubtful, it does not give you the accepted name.
Yes, I was just going to ask
Usage
that
key,
only.
which it is giving in this. So here the interpretation of this is according to GBIF, rather than using Magnoliophyta, you should be using the Tracheophyta, because that's the phylum that is accepted, right? So that is how it is interpreted. So just to understand this, so that when you are running some of your queries, how you need to be using that data. So this part is for us to just learn about that. Yeah, so these are the taxon IDs that it and this is a little bit of explanation. Okay. So now there is another way of doing this if you want to do for multiple species. GBIF has provided a tool to do that. That tool is called as species matching tool. So if you go to tools, you can search, you can select that species matching tool. And then once you open it, you get an interface like this. And what you are supposed to do is actually create a CSV file with name and preferably kingdom, and write these and save it as a you can create it using Excel or Google Sheets, but save it as a CSV file, and then you can just select from here or drag and drop here. Once you do that, it will open that file. It will give you this particular menu, and then it will give this button saying match to GBIF backbone. Once you click that, it will go and fetch those names and show you its interpretation. So in this case, it says that it is matching to these are exact matches, and it again the same status is shown here, correct? And if you want, you can go and change those. So here this magnolophyta, if you say, okay, since this is synonym, I actually want to change it. You can click on this pen button and you can change it like this. And then once you are done with whatever changes you want to do, you can click on this generate CSV button at the bottom, and you will get the whole CSV with all the taxonomy. And this also provides you the keys. So this is another way of processing multiple names to get taxon keys and other taxonomic information.
So
just wanted
can
to
we
show
fetch
you.
the entire higher taxonomy through this, Vijay?
Yeah, it is here, no? Kingdom, phylum, you see, you get the whole higher taxonomy. I actually use this tool for my data publication also. I send all
Yeah,
these.
I can see that when we just have the names, it just simplifies the datasheet creation.
That, and it also once checks your name spelling.
Yeah,
So
spellings and errors.
I always try to go through this before I use, and then just copy paste these fields to my data set so that I have the whole higher taxonomy and according to GB, which is the advantage. So this is another tool I just wanted to basically show you. So that's all for this session from me. Any questions on this?
Yeah, I have one. What is the use of that key? The key for the
So that key
species.
is internal key that GBIF uses, but
Mm-hmm.
using that key to query the data will fetch all the records with that key, even if there are slightly different spelling variations or if there are any synonyms or anything, everything
Okay.
GBIF will fetch. So whenever we are running any queries for analysis, you are recommended to use the keys rather than just the name.
Okay.
Any other
Thank you.
questions? Yeah.
Sir, just please repeat once again that how to import the higher taxonomy of a list of species, that part.
Oh, it's actually a very user-friendly tool. The only thing you need is you need a CSV file with scientific names and phylum, preferably.
Yes, because some genus have same names both for plants and
Exactly.
yes.
So if you know phylum is, if you don't know, it doesn't matter. But if you are processing, say, a list of butterfly names or a list of plants, it's better to give the phylum so that the matches are better. So you create this particular file, and in this interface, you just drag and drop it here. It will open this interface, right? And just click on match to GBIF backbone.
Okay.
It will start matching and give this interactive thing. It will give, I think, 25 per page. So if you have 100 or 200, it will give you pagination at the bottom.
And then from
And you
here,
can
we will have to generate the CSV,
generate
right?
CSV. Yeah.
Okay.
And you have the CSV file for download. And then you can open that CSV file.
So, do we need only CSV file to do this, or can we do this from Excel sheet also?
No, only CSV.
Okay.
Only CSV it accepts here. Yeah.
Excel sheet will like easily
You can
cause
save it
some
as CSV
issue.
quickly.
Yeah,
Mm.
so you can just like when you save your file, you can just save as CSVs, like text file. It will not cause any like data issues, or it will keep your structure of data file. So CSV is like, yeah, more
And
Is a better.
a safer option to
Yeah.
save your file without messing up the format.
And one important thing to note is you should have the scientific names in field called as scientific name exactly as Darwin 4. So S is small and N is capital. And kingdom, everything is in small. So once you have these, it automatically does it very quickly. Only thing to remember is you need to have a field called scientific name and preferably a field called the kingdom. And obviously you can have a field as ID so that you can link back the data in case there are too many rows.
So
Clear?
scientific name and kingdom should be sufficient. ID, I
Yes.
think, is optional. Yes.
ID, in case like you have hundreds of names, then it's better to have that ID so that your rows are matching properly. You can make sure using that.
Yes, yes, yes. Okay. Thank you.
Any other questions? No? Then back to Melissa then.
Okay. Okay, just no more questions. I think okay. Another question. So I'm gonna just say, so we can use match species tools for R. Daphne, could you help
Uh,
with?
I think the species matching tool that you just see that Vijay just showed is the website from GBIF. But if your purpose is to get the data that you see from the CSV file just now, we can also download it from R as well. They have a function for it.
Yeah, actually, like you can do the similar things
Same
on
thing.
R.
Yeah,
Yeah,
just
but
in R. Yes.
the tool is for the interface that GBIF provide design for you, and it's like more user friendly if you don't use R.
Well, in OpenRefine, it was taught in data mobilization. I had been using that for the higher taxonomy. The
Yeah,
API key and everything was provided, so just to fetch the higher taxonomy.
yes. So it's like
But
there
this
are
is
many ways
faster.
to use it.
This is much faster than even doing it in OpenRefine.
It's kind of like different purpose of use. So it's
Okay.
also depends on, yeah, your purpose and
Okay.
how familiar, which tool you are more familiar with. Okay. Okay, and I think we can move on. Next session is the introduction to RGBIF. So I'll give the floor to Daphne.
Okay, you guys are viewing my slide well.
Yes.
Okay. Okay, so hello again, I'm Daphne. So today I will be teaching you guys a really brief introduction to this RGBIF and how we can use R to download data from GBIF. But um, I'm sorry to say because of time limit, we are not actually able to teach you the basic of R because that is another whole different thing to teach. What what we are trying to achieve here today is to let you know and show you how we can use R to download GBIF data and do some basic data cleaning and then data visualization of map. So wait, hold on. Okay, so our outline for today, for the remaining of the day, is I will just give you a really brief introduction to R and then what is R Studio and about RGB package. After that, we will have a group photo and a break time. And after break, we will use to break time to help anyone who has trouble installing R and R Studio. So we will talk about that later. And then we will do, and then after that, after the break time, I will do a quick demo using the Limo Kata example, which the code you can also find it from the training material. And I will send the link again later. And then I'll show the quick demo on what we are going to do. What is the basic and general logic about the whole script. And then after that, we will try to make you run it yourself, but we will be here to answer questions. Okay, so lastly, and then we will also have Q&A session for anyone who has any question. Okay, so for people here who have already background, sorry, you have to bear with me for a moment. But I would only just also say it really briefly for participants here who have like really basic and no background about R. So R is basically programming language, commonly used for statistics and research. I realize I might be wrong, but I realize most life science, ecology and biologist researcher, we use R quite often. And then and maybe also some different biomedical field, they use other different kind of programming language. For example, SPSS. So that is also a very popular tool for statistics. And some other field they use Python. So all of them are different kind of tool for you to do statistics. So, and then for R itself, this is an open source language. And then they have also thousands of R packages, and these packages are developed by sometimes company, sometimes different organization. And Vijay here is also the core developer of the RGB package that I'm going to show you later. So we use all these different R package for different kind of data analysis that you would like to do. And RStudio is the software and interface, the environment designed to make you working with R easier. So you will have the interface later where you can write code and then you can run the analysis, you can visualize your plot, and then manage file all in one place. So this is the RStudio interface looks like. So I hope later during break time, everyone have that ready. Oh yeah, okay, basically I just showed this already. So this is where we are going to load our script later to run, and then you can do your code editing here. And these are the console where you do when you run the code. But most of the sometimes you can do it both here. So depends on the file type that you are loading here. And then we can also have different kind of panels where you can do different things. So yeah, so this is just the basic introduction. So this is the interface that we will be using to run our R script later. Okay, so RGBIF. So this is the main thing that we are going to show today. So this is a R package. RGBIF already write different many functions to wrap the structure of GBIF API. So we can run R and use R function from RGBIF to download GBIF data directly. So it's very useful if you are already familiar with R package, with R programming. And then for you to download the data, and then we also, like just now, what we did earlier, looking up species name, etc. All these kind of different things that we want to do, we can also do it in using RGBIF. Okay, so RGBIF has documentation. This is the really detailed documentation, which I find it quite nice. Is over here. I can also send the link later. This is the homepage of the RGBIF package. and then you can see how they get you to install the getting started and then the basic function. And I highly recommend you guys to take a look at these three parts. And in get started, so this is the basic thing about the package. And then they also show the most commonly used function in this page, and you can take a look. For reference, this page shows almost all function under GBIF, and then different function when you click in, it provides you example and more detailed explanation of what you can do using this function. So because of time limit, we cannot go through everything. So I highly recommend you to take a look at and explore what function do they have and whether all these things will be helpful for any analysis that you plan to do. And then in articles, you can also see these are some kind of like mini blogs that people wrote to show you the example and case study of how they use this particular function, and then how they do the how they use different kind of what do you call that, a standard or to to customize what you are going to write. So for example, this page is showing all about the maps, and then there are also different example that let you explore different kind of things that you want to do. So I highly recommend you to look at this page. They have more detailed example compared to what we are going to show you later. Okay, so before, I think this is just the really basic thing that I can show so far because of time limit. So right now we have group photo time before, and then a break before we move on to the more demo. So maybe should I? Okay, so has anyone here? Can anyone give me a thumbs up in or any emoji in the chat box if you have R experience? I want to roughly see how many of you have R experience. Okay, about half, more than half participants here have experience in using R. So I guess hopefully it's working well later, because the experience with us doing this workshop, sometimes we cannot really know the basic setup of your current computer. Sometimes we might face issue of installation problem, and sometimes not even our installation, it's just it's package installation. So yeah, okay, so hopefully we will have time later to discuss all this and and we can try to do basic troubleshooting if you have any issue. And also let us know in the chat box whether you still you have issue downloading R and R Studio. So, and then later after group photo during the break time, I can try to help whether installing, we can try to help troubleshoot the installation issue. Okay, I will be here just check out at the chat box to see if anyone has any issue. So Melissa, maybe we can
Yes.
have a group photo session right now.
Yeah. Okay, I hope everyone is back. If you're back, please give me some reaction, thumbs up, or yeah, text in chat box. Okay. Great. I think almost everyone is back. So, the last part, the whole we will have an hour of the RGBIF demonstration and exercise. So still Daphne to lead the session.
Okay, let me share my screen. Okay, so I hope everyone has already downloaded their R and done installing R&R Studio. And there's also one file in the Lemokata project. I think, is it under here? The R Markdown file. Yes, this one. I hope everyone is also done downloading this because we will be running the script from this file directly during our exercise. So I hope you get this ready as well and then load it to the environment. Okay, so before we get into before we start using R, I want to first show you the general and basic background, give you an overview of what we are going to do before we go to the R exercise. So this is currently the general picture first. So we are doing this tutorial looking and downloading for this particular species, the mocatal. And then, so this is a native species in Madagascar. Okay, so first, when we go to R later, I will also show you, I will also copy to you the script directly to install all the package. So the installation shouldn't take long time. And then after installing, we will have to load them into our environment. So this is what we will be running. So this is just an environment preparation before we do everything else. Okay, so after that, we will download world map because we want to plot our species occurrences on the map. So because I mentioned also this is a native species from Madagascar, right? So we also download specifically Madagascar's shapefile from this function. Okay, and then now we go into the most important part, downloading data from GBIF. So we insert directly the species we are looking for, and then using this function called name backbone. So the result return look like this. So they will give us what we see just now on the previous session that VJ showed. We got usage key, we know the scientific name, and then we know the status. So all of these things are the one that we do just now in the previous session. So all this information, you can also do it here in R. Okay, so why are we running this? The reason we are running is because we are getting usage key, because we need the usage key to download the species occurrence data, this taxon key. Okay, so this will be the I would say the most important function of this workshop is to download occurrence. So these are the function, and then inside here you will have to we will have to replace the usage key for this particular species over here, so we can download occurrence for this particular species, and then we will also have to mention what kind of format that we need, and then the user password and email where you use to register for GBIF account. So during the exercise later, you will have to remember to type in and replace all these things from the script later. Okay. After searching for the occurrence data from the particular usage key of this particular species that we are looking for, we will go to download them. So this particular function will download the file that the previous code found into your computer. So this is example of the occurrence table that GBIF return, the JSON return, and then using this code, it will directly run and download into your computer, and then we can use that later for analysis. Okay, so one of the important thing to mention here is whenever we use GBIF data, we have to remember to cite them. So this is the code that have already written for all of us. After we run this later, you will see they will show us how we can cite this particular download, and then they will also give us DOI for this citation. Okay, now let's move on. After downloading, we will show you data visualization. We are plotting all this occurrence data on the map. So this the first overall picture is a world map, and then the red dot is the species occurrence with coordinates. But then because I mentioned the species we are looking is native Madagascar species, right? So why are there so many red dot outside of this Madagascar? So some possibility will be these are records of zoo specimen, so living specimen or preserved specimen somewhere around the world in their museum or in their zoo. So that also considered as a record. So of course you will see a species occurrence in places outside of Madagascar. So this is the quick first overview of our data set. But then because we want to look at for this particular tutorial, we are only interested to look at data within Madagascar, right? So we will have to do some data cleaning. So most of the thing here we are running to clean this data set. So for example, we do the first thing, we filter out data that is without coordinates, and then we want to check what are the distribution, the spatial distribution of this data right now. Where are they located in different countries? And then to check the basic information from this data set. And following this tutorial, the first step of data cleaning is to look at basis of record. So as I mentioned just now, some of them may be preserved specimen, some of them may be living specimen. So it depends on the data that you want to evaluate. So we are not interested in looking at fossil specimen and material sample. In case you don't know what material sample is, one of the example is, for example, tissue sample. So, for example, this university collected tissue sample from this species because they want to extract DNA. Yeah, so this is also considered a record in GBIF, but then their basis of record will use material sample instead of human observation. Okay, so for this part of the purpose, we are not interested at looking at this type of sample. So we are deleting them. We want to exclude them from our analysis. So these are the things that we will run later and we can show. Okay, after removing this data, we try to plot the map again. And then we will see some occurrence they remove already. Maybe it's easier, it's more obvious later when we are running it from the R. Okay, so over here, step 2, we are flagging problematic coordinates. So depends on your purpose as well. Like for example, I know exactly that I only want to look at data located inside Madagascar. So I know exactly what kind of data filtering I want to remove them from my further analysis. So over here, I do filter, for example, I don't want any data that is without coordinate because they are not specific enough, right? I want only data located in Madagascar. And then so all these function, which is also quite useful, but I cannot get into detail of all of them. But you can also check out, for example, I don't want any records that is duplicated in terms of same location. So over here, they look at if you have another record that have similar latitude and longitude, they will remove them. And then for this one, for identical lat long. And then they will also, for this function, they also remove invalid coordinates and also coordinates that fall on the sea instead on the land. Yeah, so, and then for example, these are also different kind of buffer in terms of distance that I want my coordinates to be specific enough for that place that I'm going to do analysis. For example, they have also which one is the best example? Okay, so because let's use this example. This function is particular to remove and omit occurrence data that has coordinate falls on the national capital instead of the original location of that species. Because when data publisher are publishing data, there is sometimes a misplace and misuse of coordinates that they put the country coordinates on this particular data. So these are all the filter and function just to be safe for us to filter out any potential problematic data to further on for our analysis. So this is a cleaning step, particularly focused on coordinates and location cleaning. Okay, so over here, you can see this plot is to show the raw data is in black dot. It's not obvious here. So for example, some of these, because they already run the data cleaning on the coordinate cleaning functions, right? We already removed those data that is problematic, so it's recorded black here. Those data that pass the quality check will still be red. So you can see now, okay, so maybe right now, all the red location is the one that indeed we want to filter. It falls on Madagascar. Okay, so now we don't have to look into the world map anymore. Let's zoom into Madagascar and then look at the species distribution. Okay, so step 3 part of data cleaning is to look at the quality. Because sometimes if you have experience in doing field work and depends on the GPS you are using. Sometimes some GPS have really high precision, but sometimes you use your old handphone for GPS recording. So this thing may create error or bias into this occurrence data. So to be safe, you want to filter out all these data they have uncertainty. So specifically for this example, this step it remove coordinate uncertainty in meter that is more than 10,000. So if it's more than 10,000 meter, they remove it from the data set. Yeah, so this is the coordinate quality checking step. After that, we do a plotting again just to see how much we have removed. Okay, so the black dot is the one that didn't pass our quality check. And then the red is the one that still has good quality, so we keep it. And then the next step is temporal. So for example, right now you are interested to look at data that is after 1955. So this is the year that you set because you only want to look at data after this year. So you will set the filter for this and do the cleaning again. Also, that gives you the remaining data. So as you see the different cleaning step by step, the more higher quality threshold that you set is going to remove more data that you expected. So it really depends on your purpose of analysis, but because you only need to, for your analysis, you only need to use those quality that is good for your work, right? So sometimes it may remove a lot of data. So sometimes you can also adjust the filtering criteria differently. Yeah. So, okay, so this is just the really basic way of using RGBIF. And we are here showing you some of the general steps on flagging and cleaning all those data that may have quality issue. So these are the things that we are going to do later. Then we can all run it together in R. So right now I will stop sharing my screen for a moment before we move to R. And okay, so anyone has any questions so far before we start our practical session? Okay, looks like everyone understand what I say.
No. Actually, we
Okay.
have to do hands-on, then we'll understand it better. We need to do hands-on, then we'll understand better.
Yes. We will do hands-on right now. If no one has any question, okay, I will be resharing my screen and we can do the hands-on activity all together. And then if Melissa and Vijay is here to also look at the chat box, and you can also feel free to ask any question during the hands-on practical session because we are all going to do this all together. Okay? My
And
screen.
yeah, please just open your R Studio and then follow Daphne step by step. And if it's too fast for you, please also let us know.
Okay, so for people that has no experience in R yet, we will have to install all this package first. I wrote this already for you. You can copy paste in your R. Okay, so let's go to, okay, this is the R Studio interface that we are going to run and do our hands-on practical. And then we have to load our R Markdown file here, the file that I mentioned just now. Has anyone has any issue loading it? And please
How
voice
do we
out
load
or
this project? I mean, the file in the R Studio, Daphne? Because when
sorry, what kind of R?
yeah,
Mhm.
the Limakata project file, what was given to us, when I am opening it, it is opening in GitHub. How do I get it from
Can
there?
you click open file here and then
Okay.
to load it into your R Studio?
But the file, what I am, when I'm opening it from the original source, what was given to us, it is getting open on the GitHub, I think. I am not sure what is happening. Till yesterday, it was opening there.
Maybe because you haven't downloaded R Studio yet, is it? Because I think
Open R Studio and then open the file there.
I have opened the R Studio.
yes,
I am
yes,
From
now
R
opening the
yes.
file.
Studio, open file.
So I'm getting a page. where I can see things, but I don't know. I mean, yeah, I can see the text kind of thing. I'm not sure
Ah,
what it is. That right?
you
But it
want to
is on
Well,
share
the
screen?
browser. On the Chrome, it is opening.
No, no, no.
It is
Can you
showing. It is showing like this in my case. Uh, Colorations in the text and not showing.
Yeah, correct. This is how it should show.
but you
Okay,
load it as
you
a
have
text
the,
file?
yeah, it has opened as a text.
And how do
Can
we install these things? What Daphne just pasted? Install packages?
can you? For this for this particular R Markdown file, have you already opened RStudio like me?
Yes, I have
Are you
opened
on
the
your
RStudio.
okay? So
Yeah.
go to open here,
Okay.
open,
Hmm.
look for your RMD file,
That is what I'm saying. The RMD file is not
and then
getting
click open.
downloaded. The RMD file is not getting downloaded. It is just opening in the browser.
Daphne, can you just share the file in chat?
I can share
Because
the file.
when people are downloading the file, probably they are getting a
I can also
.txt
share.
extension at the end.
This file,
Yeah,
I can also share. But
the
wait a
RMD
minute.
file.
Let me share it.
I
Yes,
already put it in the chat box.
yeah.
You already did?
So Aditi,
Okay,
just try
because
Yes.
downloading this
Yes,
file.
yes. Now it is working
Okay.
fine.
it
Yeah.
also did
Now it is getting downloaded. Yeah.
So after you
On
download, you see if it is. So um,
Actually,
it
that
should
was getting
show.
downloaded as a TXT file, not as an RMD.
Exactly. That was
Yeah,
what I was
please.
typing, but then I realized and I just. So
Yeah,
once you
please
load it in
make sure the
RStudio,
format is
Yeah,
Markdown.
now the
Yes.
RMD file is downloaded in the is opening in the RStudio,
and it is
and
showing like this colorful, no?
colorful. Let me check. Yeah, blue green colors I can
Right.
see.
Because
Okay.
what Subhajit shared earlier was black and white, and that was
Yeah,
not the right
no.
format.
I can see that, but I am getting a flash saying that packages, coordinate cleaner, country node, and others are required
Right.
not installed. So
Yeah.
how do we install the packages?
If you
Daphne
click on
just
that, it will install it.
okay.
If
You can
not,
click on the
Install. I did that just
you can also copy paste my install packages code in the chat, and then they will install all the packages directly for you.
That is what I was trying to actually
You,
do.
you
I
have to
was
you have to copy and paste on console, not on the project window.
Yes.
correct. In the
Yes.
console window
yeah,
now.
but as soon as I click the install, that thing which was showing above the
Yeah.
this thing, it is automatically downloading it.
Installing it.
Okay,
It
so
takes
for people
better.
have no experience in R, please copy paste this thing into the console part and then click enter so that they will install all the packages like what you see on my screen.
Yeah, it's done. Yeah, it installed everything.
Yeah, so
So
all
I
these
have a basic
package.
query since I have not worked in R. So do we have to do this installation every time we work, or it is the one time thing that now it's installed? So next time when I work, I need not do that.
No. So you already install all this package in your document.
Okay.
So, in your computer. So next
Yeah.
time you
Okay.
only just have to run this library and call them
Okay.
out. You don't have to install them anymore. So this is just a
Okay.
one-time thing. Mm.
Okay. Yeah. Thank you.
But you might have to install new packages all the time. This particular file is using these packages, which is
So whatever
RGBIF
we
and
will need, that has to be installed every
yeah,
time next. Yeah.
yeah. And if there is a new version of package available, that also you will get a notification.
Okay.
There are some packages which are renewed every few months, depending on the new functionality.
Okay, so I will carry on. Okay, so after you install all of them, you should run this library. Over here, you can see a green play button on the top right of this code script. You can click run. So they will run and load all these packages into your current environment. So after that, oh, I forgot to mention just now, the tutorial that I was showing and explaining earlier in this page is actually the exact same thing. This thing that I show you just now is actually the exact same thing we are doing here in the Markdown file. Because I'm using this to explain because it looks quite nice. It's more difficult to view at over here. Yeah, so we are running the exact same thing that I just showed you. Okay.
Definitely, we need to run the selected lines or the whole code.
Yeah, that is what even I was asking.
Sorry, can you repeat the question?
Do you need to run the selected lines, that library packages, or run the whole code?
The green small arrow
When
on
you
the right
click
of
this
the
It
green
is
play
too.
button,
It
they
is a
already
drop down. It's a drop down menu giving us
No,
options:
no, it's
run
okay. That's
lines
okay. But whether
or
you have to run the
run
whole
current.
code or only the selected lines, there are
uh,
options there to run the
right.
selected lines or run the whole code.
but when I
See,
click this, it's already running everything.
We have to run the whole code because the other
Yeah,
things
run everything.
are
Yes.
they
Okay.
are mentioned as comments only, so they will not run anyway.
I want to talk something. So there are two ways you can run this. There is one run button on the top.
This here.
Immediately. No, on the top, little next to
Here,
public.
this.
So yeah, don't use this right now. Right
Okay.
now, what you are using is every code block. Every block that has our code has a little green button.
Okay.
That we are using right now. So that
Okay.
we are in some sense debugging. We are going step by step.
Okay.
If you are
Yes, we are going step by step now.
yeah, if you want to run it completely, then you have options from top. But currently we are running these small green arrows in each code box just to go through one one set of code lines at a time.
Now we have installed the
No,
libraries.
I did not follow very clearly. I could not get it.
So there are 2 or 3 ways we can run this code. Okay.
Mm-hmm.
So one way is using those little green arrows that are
This here.
yeah, here. What now Daphne is pointing to.
Yeah.
That is what we are going to use right now. On top
Okay.
is another run button, which has
Mm-hmm.
different drop down options.
Option.
We are not
Okay.
going to use that. The third
Okay.
to it will be another option NIT, KNIT, under the Lemurkatta project RMD file. There
Mm-hmm.
are few more options to run that. So there are different ways of doing it. Right now, what we are going to do is run each of these
Step
code
by step.
by one
Yes.
using that little green arrow
Okay.
next to each code block. Correct?
Okay.
Is it
Okay.
clear now?
Got it. Yes, yes, it's clear now.
Okay. I will move on because I scared it's taking a lot of time. Okay. So after we load all these things into environment, we have to load a world map into our environments as well. So run this thing. And you can also when you move your cursor a pointer here, you can also click enter and they will run as well. Over here you can see. Okay, so after world map, and we are also downloading Madagascar map, so we can click to download.
One more point. Once you run this in the bottom, your cursor should come to a new line and stop there. Should not give any errors. Like totally at the bottom of the
Yes.
screen, if you see, her cursor is on the new line after 2 pluses and all. So that's where if the cursor is, that means the lines are already executed and you are ready for the next command.
Okay, so move on. After downloading the map, now we are going to look at the species we want to download. So if you remember, this is the exact thing that I showed you just now, right? We click run. So this part, we are viewing the information about Lemokata because we want to get this usage key. So after we get the usage key, we can assign it to usage key, run it, so that later when we do our occurrence download, we are downloading data for this particular species only. So there's a lot of text here, but I cannot go through them one by one because of time. I highly recommend you to read it when you have to, after this workshop, so that you can get the detail about everything. So all these text I cannot go into detail, but please read it later so that you can get a better pictures. Okay. And also for this example, right now, the practical we are running, we are still running the one single species only. Okay, all right. Okay, after getting usage key, and then we assign it here. Now that we know that we are only looking at this particular species. Now let's move on to occurrence download. Line about 84, 85. So here, please help me to insert and edit your username, password, and email for your GBIF account. I give you 2 minutes to complete this. If you remember the username, the password, and the email you use for registering GBIF account, this is the place where you insert them. Please do not.
I have done
Yes.
this. So should the dataset come to me as a mail?
Sorry?
After running this, will the dataset the CSV be mailed to me?
No, not mail. This is just an authentication access
Okay.
to let GBIF
Yes.
know, okay, this particular person is downloading. It's
Understood.
not, yeah,
This is for
okay. It's not
security
mailing.
purpose.
Yeah, yes,
Understood.
exactly.
Ask for permission that you want to take the
To download.
data from GBIF.
When I'm running this, it is showing error.
Did you upload your? Did you write your username, password, and email?
Yeah,
correctly?
I did that. I replaced the usage key also. We have to replace the usage key, right? We have to enter the
No.
usage key.
No, because we are already
Not here.
assigning usage key here.
You are not supposed to.
Did you run this code already?
I was trying to run it. I entered the username and all. Maybe that's why it's showing error. Yeah, now it show. No, it still says error unauthorized. I don't know why.
Okay, your Gmail password has to be put.
No.
If you're logged into Gmail.
Oh.
If I'm logged into Gmail, then
Your Gmail
What is the error you are having?
it is showing error 401 unauthorized. I don't think there's any.
Possibly wrong pass.
No, I just logged in with the same password, so that it cannot be that.
Or maybe you
I'm not
can
sure what kind of error is that.
upload the
401.
screenshot.
Yeah, should I share the screenshot?
Yes.
Yes, please.
Okay. While you share the screenshot, I should move on. So, after anyone already insert your information and run this code,
Why I am also getting that username? Why there's an error?
Is only some of you having that error, or only 2 of you so far?
I'm also unable to log in because I have logged in through my Gmail.
I have also logged in through Gmail only.
I'm not understanding what exactly. I have just shared the screenshot.
GBIF. Let me log in एक बार. What is my Gmail password?
We have to put the Gmail password or the
Yeah, I
GBIF
think you have to put the
password.
Gmail password. That's the password you have.
Oh my God.
No. GB password only.
No, it is a GBI
GB,
password.
GB password.
Yeah,
Yes, GB
Yes.
password.
there has to be GB
GBIF
password.
password.
GB
But if
password and
it's your
GB username.
Gmail, if it's your Gmail password, don't share it here.
Let me log out. The problem is.
Was anyone able to do it or everyone is getting it?
Just a minute, let me.
I was able to do it.
Okay, so Damini was able to do it and Subhajit was able to do it.
I'm
So there
not
are
sure.
seven
I'm
people who
logged
have.
into GBIF with the same password, so I don't see the reason it is showing.
Yeah,
Can Vijay help solve this issue in the
yeah.
chat box while I continue my lesson?
Yes,
Okay.
Yes, please.
So for anyone that has successfully
Yeah.
running this, okay.
We can reset the
Now
password
this
and go
step,
GB.
yeah.
so now this step is
But
to
one question for me, no error is showing, but a download download ID or a download data set, something like that is not
Wait.
showing.
That will come. Did you
okay.
try the wait command?
What command?
100 no, I'm talking to Subhajit.
Yes,
Okay.
Subhajit, 105, have you run that?
No, not yet.
Okay. So once you run
This
that, you
is
will
the
know.
part. This is the part I'm going to say later.
Okay.
Right now. Okay. So for people that have unauthorized issue, please discuss it with VJ at the chat box while I continue the lesson. Okay, so for
Yes.
anyone that has no issue on logging
No
putting
issues
your
now.
authentication, now we have to run this occurrence download wait, and you can run everything. So this is the part where you really download the occurrence data from GBIF. Occurrence download get is the function where we can download. Yes, we will get the data set. So this should take a while. And let's see. Most of the time, if the data you are query is a huge data set, so this sometimes will take a while. You will have to wait for it because it is a huge data set, right? So this example we are using is not a big data set, so it should be really quick. And here, if you see this download file size 0.09 MB, that means we already successfully download it. And this data set is now in your computer already in this file path. Okay, now that we got it and we downloaded it in our computer, this part is to show us the citation. And GBIF are giving us DOI, over here. And then they wrote it all, format it properly for us. If you downloaded this data and you use it in your paper or in your reports, you can cite this citation directly. Here is the DOI as well. And then even have the date ready. So this particular code is for you to get the citation so you can use. Okay, now let's move on to data visualization. Okay, so this species we are looking at is native to Madagascar. So, but because we are downloading everything, right? We're downloading raw data. So now let's run this particular code first to look at everything, like the world distribution of this, the occurrence data so far on the world. Yeah, so like I mentioned just now, because we only interested with data over here, right, in Madagascar. So along the cleaning step later, we will be removing all these steps outside of Madagascar. This is just a quick overview so far. Okay, let's move on. So this is we are also running this particular step later in one of the cleaning step, but this is just to quickly filter away those data that has no coordinates.
Okay.
And then for this code in line 151, this is to show us how many data right now in which country. So you can see there's one of these species in China, 3 in Denmark, and then 783 in Madagascar. Now, this is the overview of the data set we just downloaded. And we are also interested to look at, because like we mentioned, they are records outside of Madagascar, right? So they can be maybe museum specimen, they can be maybe living specimen in the zoo. So how can we know that we can use this basis of record to look at what is the record type? Yeah, so yeah, you can see some of them are actually preserved specimen. Some of them are actually material sample, probably for DNA extraction work. Yeah, but we would like to exclude this from our analysis. Okay, so establishment means this is for looking at records of this species. So for example, because not many people use these Darwin core terms in the while they are publishing the data to GBIF, so there's not much data about it, but it's still a good overview for you to look at because it happens that this Lermocata is native, so it's just also a quick overview to look at your data. Okay, now we move on to data cleaning step. Okay, step 1, we are removing data that is fossil specimen and material sample. Okay, so this is what I show just now.
25
We run them.
Yeah, 25 records are removed.
Okay, so yeah, the first cleaning step let us know that 25 records are deleted. So possibly that 25 records are some of them are come from this category which we do not want them. Okay, so after this cleaning step, we know that there are 811 records remaining for our following. Now we move on. Before we go to the cleaning step, let's put it on the map again to see where is the 25 codes located and where are the remaining steps. Yeah, you can see this particular record from China, and then from 2 or 3 from the US. They are not part of our interested data, so we remove them already. Okay, now continue. So the cleaning step 2 is what I said just now. This is focusing particularly on filtering data that is about problematic coordinates and location. Yeah, it can be duplicates, it can be zero coordinates, it can be data outside of Madagascar. So this step is more convenient for us because we already know we want our data in Madagascar only. Okay, so we run it. Now we see 124 records deleted. Yes, okay. So these are the data, 124 records that does not fulfill our quality check. Okay, we plot them again. Okay, you can see because few of our filter, because we only want Madagascar, right? You can see all the points outside Madagascar become black dot already, which the data we do not want. Okay, so looks like the cleaning step are good so far. Now we can zoom into Madagascar now, because this is our interested location, right? Okay. These are the dot so far, the distribution, and then we have to do the cleaning step now. Okay, now this is about coordinate quality. So like I mentioned, I do not want any species record that has uncertainty in coordinates more than 10,000 meters. So I use particularly these terms to filter them out. So I run them. Okay, so now we remove a lot of them because they do not fulfill my quality filter. So 800 something records deleted and only 22 records remaining. This is actually quite common if you use GBIF data and you have really high standard on the data quality. It really depends on the analysis you are doing. Okay, so you will have to decide the quality filter. Okay, now we run. Okay, so again, the black color dot represent the data that is not fulfilling my quality threshold. So these are the data that I'm going to remove them. And then the red color dots are the one that keep remaining for my continue the future analysis. Okay. Now that we complete the cleaning on the location part, we want to filter data that is year after 1955. So this step is to filter year. Okay, now we only left 7.
7 records out of so much of.
Yes, exactly. So that means a lot of records actually do not fulfill my quality standard. Okay, we plot them on map again. Okay, so the remaining red dots are the data that really fits my good quality standard. And then I will use them for the analysis that I wanted to do. Okay, so this is actually just a demo and a really simple basic example of what data cleaning can do to GBIF data before you use them for your analysis. So not everyone uses the same quality filtering. It really depends on what analysis you are going to do. And some of the, for example, let's go to this step. This function comes from the library, the package coordinate cleaner. Wait, let me here from coordinate cleaner. So you can also go check out their documentation because they have a lot of function for you to do data quality check and cleaning for your GBIF occurrence data. And yeah, so these are some of the example that I can show you today about quality check, about downloading, about cleaning, and then plotting the data set. So yeah, I hope this is a good basic script for you to start
Definitely one
using
question.
GBIF data. Yeah, okay.
Yeah, definitely. After you clean the data, that data will remain in the on our interface. I have to download the data again as a CSV file, that filtered data, the
Good
clean
question.
data.
Yeah, it came to my mind too. Yeah.
You want to download the clean data.
Yeah.
Use write.csv command and save it. So that's where we'll have to start learning R a little bit.
Yes, yes, understood.
Okay.
Yeah, this is the part of basic you have to learn.
No, that I understood. It's that's simple,
So
simple
it
concept.
will
Yes, yes.
automatically be downloaded as a CSV or in our download folder
No, you have to
after
download
this.
it after cleaning.
Any
Will download it.
after writing this write.csv
Yes,
step. Okay.
yes.
So
Yeah.
as we run it, it will be downloaded in the.
Yeah.
Okay, okay.
I
This
had
That
one more
command
question.
basically
Oh, yeah.
tells that whatever data you have in clean step.4, write it as lemurkata occurrence clean CSV. That's the command we are giving here.
Okay, I had one more question. I forgot, but what it was. Ah, yes. What I was about to ask was now whatever this RMD file has been given to us, we can use this as a basic structure or framework to try and practice it for on our studio, right?
Yes,
Mhm.
exactly.
Yeah. So my question was, Daphne, that if I want to make some changes according to my data in this basic draft, how do I
Mm.
do it and save it as a RMD file?
Uh, it's like you can use save as. For example, this is the structure that you are using it right now, right? You
Okay.
can, for example, let's say you make some changes.
So
You
here
are looking,
itself in the interface, I can make the changes.
yeah, here.
Okay,
For
okay,
example,
okay.
you are
Yes, I
looking
got
at
it.
this species, yeah, and you have to. It's like you are writing your Word document using Microsoft Word document. You
Yeah.
click save as.
Save as .rmd.
Yes, you click save
Okay,
as.
that is
And
what I
then,
wanted to know.
yeah, you
Okay.
click save as and then you're saving. Okay, now
.rmd.
this is a
Okay.
different project.
I got it.
What is
Yeah.
the actually? Okay. What should be the that code again? We just show that code that Melissa that is what that code you have written that at the end for that downloading.
Means saving.
Saving file
Yeah.
is write.csv.
Uh-huh.
And when
Writer
you write,
CSV step 4 lemur katta occurrence clean row
so
dot names is
basically
equal to false. Okay,
you
ठीक
are
है.
telling first variable is the
हाँ,
data frame, second variable is the file name, and
okay,
row
okay.
names dot false you are writing so that it doesn't write those 1, 2, 3, 4 as
Okay, it
numeric
doesn't
number
come.
again. That you don't need for every
Nah.
record. You don't need a
In
number.
yeah. Okay.
Okay, so we now have the Q&A session. So for anyone who has any question, any issue, we can use this time to discuss. Feel free to ask question in the chat box and directly to us. Yeah, so this particular Lemokata file is a good base info for you to start working on using RGBIF. So feel free to modify it. And then of course, because this function has a lot more, this RGBIF package actually has a lot more different useful function, like I show you in their documentation. Feel free to also check them out because this might be useful for your analysis. Yeah, so some of it here, some of it should be really useful for your analysis as well. And because we cannot show it everything. Yeah, so feel free to take a look at the documentation and also check on the case study on how people are using it. And then you can do the editing in here as a start. How can I edit for multiple species in multiple country? Okay, wait, hold on. Do we have additional exercise for this part of the, let's see. Okay, here, we have exercise over here.
Okay, please look at my screen. This is the part where I can let you try to do it yourself. And using the current code in GBIF in the R Markdown, can you try to do for this species located in South Africa is a preserved
Yeah, I think
specimen or human observation, has coordinate, and then no geospatial issue. Please try to do this using the occurrence download function. But before that, we have to find the taxon key, right? So please run this to get the taxon key so we can put the taxon key in our function.
Because Saxatilis. I want to go backbone.
Yeah.
Lepus Saxatilis.
I should. Ah, anyone has successfully tried this occurrence download
doing it.
exercise? Okay.
Please show me a glimpse of the previous slide. Once.
This slide?
Yes, yes. Okay, fine,
Okay.
fine. Yes.
What is that next? The code for downloading you have to take. Okay. Lemma कट हो सकता है.
Yeah, I have copied the question here. And for those who already get the idea on what to type, these are the answer for this exercise. So when you run this, you will download the data set for this particular species and this filter that we want.
I'm not sure there is something wrong. I'm still getting, when I'm plotting, I'm still getting the plotting for the Limerkata. Why is that so? I'm not sure.
Where are you plotting?
How we did in our studio, the same way.
So you have
Did
to
you
run
verify
each and
the
every
name
step.
badge?
You
I
have to
have to.
run each and every step and make
Yeah,
sure that
I
you
did,
have.
but do I need to save it again?
No, you have to copy the usage key also in the right place. Have you
I
done
did
that?
copy the usage key. I
Of-
changed the map also from Madagascar to South Africa.
No, what I'm just give me one moment. Let me check.
For this particular exercise, I'm just showing you and want you to practice
Okay, I
on
got it. I got my
Line
error. Yes.
24.
I
how
got
to use
I
the
got it.
download,
Yeah.
I
not
got
to
it. Where
run
I went.
everything. So for those who successfully download this, anyone has any issue so far?
1 second. We're downloading.
Yeah, it is downloading.
Yeah. Yeah. Basis of record.
How many data from this particular data set?
It is still running for me. It is not downloaded yet.
Oh, it takes that long?
I don't know. For me, it is still saying running.
See.
So this data set for this exercise 5 has 15,002 records. Vijay, are you here, Vijay?
Yes.
Someone was asking, I just realized I don't have example for editing. Yeah, you replied that particular question. How do I edit for multiple species in multiple country?
Yeah, I
His
mean,
question
there
is
is
for downloading, right?
multiple species in. Yeah. So you can add multiple taxon keys,
Yes.
and
You have to
yeah.
you can use a species matching tool or just now the function, what was it, name backbone, to check or to get all your taxon key for all the species, and then use the code script just now to
Just replace the taxon key.
Your spatial issue.
Yeah, so just we have to replace the taxon key every time whenever we run it for the new this thing, right?
Yeah, for the species you want to download.
Can we try something of ours now? Still running. It's not showing anything to me.
I think
I don't think
everyone
it's
has
good. I think
No, no, no.
everyone has
sometimes
submitted a
No,
script.
it got no. It got it.
Yeah. Since so
yeah,
many of us
okay.
are running different scripts, all the queries are going to GBIF server.
Yeah,
And
I can see it in South Africa now, but outside the South Africa too.
Ayo.
Okay.
Now,
So, sorry, because of the limited time, and we may only have like 5 more minutes for questions. So if you, you can just take time.
Anyone has question?
Yeah.
and for more questions, Thursday 12:30 India
yeah,
time.
12:30 Thursday, 12:30.
Okay, if you have question, feel free to put it in the chat box, or you can stay later after the workshop and I can try to answer you. And I want to wrap up with a little summary for this part of the workshop session. So, as you know, just now at the beginning of the workshop, VJ tried to introduce you API, and then I introduce you about RGBIF, right? So if you're familiar with R and then you are already doing a lot of data analysis using R and statistic, so RGBIF is actually very good and convenient package for you to download data directly, so you can analyze and clean them. It's quite straightforward. And when you got familiar with all the function that you can use, you can actually do more complicated query compared to when you have to type all the API functions and key in the GBIF URL. So that is actually more complicated and more error prompt. So in RGBIF is actually maybe safer because you can look at the mistake quite easily. And it's also good for big data set. So you can run it and then process it directly. And it, like I said, it's streamlined to your own analysis because it's already downloaded in your computer and in your R system. You can run statistic and do some significant testing straight away. And then the code, if you're publishing paper, so most journal now actually require you to open and share your code. so that your work is reproducible by other people, and also for you, for future so that you remember what code that you wrote for this analysis. So for summary, using RGBs is a good thing for you to work around on this analysis and downloading. It also keep track so that you can refer back later. Okay, I think that's all from me, Melissa. You can take it from here.
I had a question, definitely.
Okay. Yes.
Yeah, the maps what we were mapping just now using ggplot,
Mhm.
how can we download those maps directly and use in the articles?
Yes, you
Yes, that's
can.
the purpose.
How to do that?
Uh,
You need to
I
learn
know it might be very basic, but for me it's not. Yeah.
Get more familiar with art.
Yeah, I have to. I have to.
It's actually very I think it's actually very easy to learn programming language now that we have AI. Because during my time when I was learning R, we don't have AI. So it's
Okay.
more of a typical, it's a steep challenge for beginners before that. But right now you can ask AI straight away. What
So
you
I can ask AI to give me the code to download that map.
Yes,
Is that
and it
okay?
so
But the
chatting with
thing
AI.
I did a bold connector training session last year, and what I tried to using I tried doing the same thing what you're saying right now, Daphne. But I can say that what I experienced what was, since I do not have any basic background, I could not do any troubleshooting if it is not if it is the code is having any kind of error.
From my experience, I do
Yeah.
my troubleshooting with AI as well.
Okay.
AI, you copy the error inside AI, AI will let you know what is the potential troubleshooting mistake.
Okay. Okay.
Yes, yes, yes. So I think this at this current moment,
Mhm.
it's actually easier and faster for new beginner to learn programming language now that we have AI. because
Yes.
because it gets you the answer more quickly and you can learn faster. So I think it's a good timing right now to learn the
Okay.
basic of programming using
Yes,
AI.
absolutely.
And
Yes.
it's also friendly, more friendly for like the people who cannot do the programming.
Yeah.
Mm.
Or
And
who do not
actually,
have computer science background.
yeah, and
I'm
actually
also
this,
not, I
yeah,
think most of us
RGB
here
pack.
ecology's background.
Yeah, sorry. Yes. Just want to like conclude that the RGB package is for the basic use that if you don't know how to create your script on R yourself, that GBIF can help you to just use that package, copy paste, and get the basic of the data download from the gbif.org. So it's one of the ways that you can use on R. So there are many packages you can explore yourself after the workshop.
Yeah, sure. Thank you.
And okay, let me before we have more questions, it's like let me introduce one more thing that might be also helpful for you. And let me share my screen. Okay, and before that, it is a data use
we can
club. I
we
already
can see your
paste.
Slack, Melissa.
Oh, sorry. I just like chose okay, different. Oh, okay. Yeah, here. Is that the right one?
Yes.
Okay. Yeah, so it's what I want to introduce you is Data Use Club that is kind of like learning platform and resources and community that help you to learn anything, like many things from about programming and you can developing the skills in data use. So there are three parts of the Data Use Club. The first one is training seminars. They will have like quarterly webinar series like to introduce about something, some specific topic about using GBIF mediated data. And the second is like practical session, also kind of quarterly webinar series. So it's like focusing on developing the informatic and data management skill. Also about the using GBIF mediated data. And the third one is data camp online training. It's like really useful platform, learning platform that if you that provide many courses for different programming topics like R, how to start with R and Python and many other things. You can exploit on that platform. Uh, okay, and let me show you. This is the event that about any like webinar, and you can join if you want. Just like click it and register, then you can join. They will like set the time. But so yeah, the latest one, oh, just finished. It's like 11 March. Okay. And there are like also all the practical sessions, they will provide the recordings and resource here. So like if you are interested in any of the topics, such as the introduction to API, then you will see the video on here. Oh, sorry, my internet might be a bit weird. And then when you scroll, you will see more resources about this topic. Okay. And oh, okay. Why my internet is a bit weird now? Okay, and the third I want to introduce you is, well, wait a minute. Holy. Can you hear me well? I'm
This
not sure if my internet
yes,
is
pretty clear.
okay.
What is the page you are going to show?
I want to click the previous page, but it doesn't work. No.
I think you
So
you open a new window. Maybe this page crash or something.
yeah, now every move on my website is like quite super. Okay, now it's like super, super slow. Okay. And okay, the third part is data camp. You can like GBIF like it's like collaborate with data camp. This is I think it's a company data camp donates. And then if you are a partner from GBIF participant notes, or you can apply for the license. It's like actually need to pay, but GBIF will pay for you if you are the partner from the GBIF participant notes. You can apply for the team license. Okay. And if you are individual, but you are from countries like where the GDP per capita is less than 13,500 US dollars, you can okay. You can check it here if you are not sure. And then you can apply for individual license. Okay. Then you will have like different topics you can learn from the start to advanced skills of R or of Python, and when you finish you will need to do the assessment, and when you complete the assessment you will get the certificate from that. Okay, so it's kind of like helpful resources you can learn the programming skill from. Okay, any questions about the data camp and all the sessions today.
You have just shown us the tip of the iceberg.
Pardon?
I said you have shown us the tip of the iceberg.
Yeah, let me try.
Yes, a little bit. Yes, exactly.
Very true, very true.
At least it's a start.
Yeah, at least it's the start. And
Yeah, absolutely.
I'm trying to logging in my data camp.
It is not
Okay.
showing. It is disabled. No logging is happening. It is unable to log in. It is not going to the login key, login window.
Is the link broken?
Not link broken. They might have disabled it for some purpose.
Which link?
That login of the for the data use club.
Huh. So maybe I think it's maybe try again because I'm not sure if it's like temporarily broken or not. But mine works. And okay, I can also share another thing is like in the just check show you the interface of the data camp. I just logging successfully. Here, can you see the interface of data camp?
Okay.
Yeah.
Yes.
Yes, okay. You can like do the learning and you can like go to the course you want, or there are many courses that you can learn, like Python, SQL, understanding data science, AI, machine learning, and others like Power BI or something else. And then when you like if you want, I just completed one course, and then you can go to okay, start it. Okay, and then it will have the series of the lessons you need to complete. So whenever you complete all, like there are many like some it will introduce some basic concept by via the video, and then you will need to you know you will need to do some script practicing simple exercise, and they will have the course online, so it will show you what kind of topics you will learn from this course. And when you finish it, you can try another one, and you can get a certificate from this course. Okay, so it's really helpful actually, and it's free for GBIF partners. And yeah, just try it.
So we can also log into any and do the courses free of cost, or is there some payment has to be paid?
Now you need to apply for a license.
Okay.
Yeah.
Okay.
And they will give you the license because the license actually need to be paid if you are not from the GBIF network.
Okay. Okay.
Okay. Any other questions?
If you get any questions later on Thursday, you can always join office hour and we are there.
Yeah.
And
We
the
can
details
discuss
are
more about R and something else.
yeah,
API.
and the
And
details are already shared in the email that was sent to you. The timing and the
Sorry,
Zoom.
I have a question.
Yes.
yes.
Hello, can you hear me? I saw that there is a cloud computing services offered by the GBIF, and I'm wondering if you ever have experience running any data analysis in this cloud computing service, and I'm wondering if it's only be can only be used for running GBIF related data analysis or something. I mean, I'm wondering if I could run like phylogenomics related studies because I'm a student, so I'm wondering.
I'm not sure if I understood your question. The cloud computing webinar is going to, I mean, it is talking about how you can use GBIF data to do some analysis.
So it's not like you're getting an access to the HPC something. Relate.
No, I don't think so.
Okay.
I think that is another way of accessing GBIF data, but for different purpose. Maybe it's more for the developers, right? Or for any kind of software or website rather than ecology analysis. I think if I'm not mistaken.
Yeah, thank you.
Okay. Any other questions? Okay. Since we don't have exercise and homework to complete after the workshop, and we will check all of your attendance these 2 days, and if you both attended, then like GBIF secretariat will give you the certificate. So, okay, also thank you all for staying with us for 2 day sessions. And it's like I know some of the courses is a bit like brainstorming and burning. And like if it's still I feel difficult for you, just welcome to join our Asia office hour every Thursday. Okay, then it's time to say goodbye. And thank you again for your participation. So,
Thank you.